Okay, welcome to Let's Get to the Marks. We're looking at a Biology A-Level ATP question pack walkthrough for AQA, SNAB and OCR, and we're dealing with the ATP today. That's today's topic. ATP, of course, adenosine triphosphate made up of a nitrogenous base called adenine, made up of a ribose pentose sugar, and three serially bonded or covalently bonded phosphate molecules. Awesome. So let's get into the questions. Remember to pause the video, have a go, and then I'll go through the answers. Okay, let's get into it. Water is used to hydrolyze ATP. Name the two products of ATP hydrolysis. What does hydrolysis mean? Well, hydro should remind you of hydrogen. It actually means water. And lysis means breaking. Think of lytic or lytic enzymes or lysosomes that carry digestive enzymes that break things down. Okay, so lysis, like photolysis, all sorts of ones, but it means breaking down. So we're talking about breaking down ATP. And of course, we're using a water molecule, hence the hydro. Now, so the two products are actually ADP and PI, but you can use those abbreviations and they're fine with that in the mark scheme, or you can call them by their correct name, but just be careful of the spelling. It's adenosine diphosphate. It's not adenine diphosphate, it's adenosine diphosphate, and it's inorganic phosphate. And let's not forget, there's also an enzyme involved. Do you know the name of the enzyme? It's ATPase, which is a slight abbreviation of ATP hydrolase. So suggesting that it's involving water again, ATP hydrolase or hydrolysis. And don't get that mixed up with ATP synthase that actually joins ADP and PI together to make ATP. Yeah, so there's your two answers, ADP and PI. And you need both to get 1 mark in this question. What stingy examiners? Okay, let's move on. This is the next question. Describe how an ATP molecule is formed from its component molecules. So pause the video and have a go now. Now I'll go through. Describe how an ATP molecule is formed from its component molecules. Okay, so how is ATP made? Well, we know it's made from ADP plus PI to ATP. Now, this would get you 1 mark, but it's actually a 4-mark question, and this would only get 1 mark by saying that adenosine diphosphate and inorganic phosphate make ATP. We need to actually describe how it's composed. So we've got some other things here. We've got a water molecule being released, and we've got that enzyme. Do you remember the name? That's ATPase. Um, so sorry, ATP synthase. Just got myself in a muddle there. ATPase is the one that breaks ATP down. So ATP, what's it composed of? For this type of question, I'd describe exactly what it's composed of first. So I'd be like, ATP is made of adenine, which is a nitrogenous base, if you wanted to add that, a pentose sugar, which is ribose, and 3 phosphate molecules. So we've given a nice description of ATP. Now, how is it joined together? Well, in a condensation reaction, water is produced when ADP and inorganic phosphate are joined together by ATP synthase to make ATP. So I just get all of the information down in a nice described question like this. It's just a case of the examiner's going to read through and just check how many facts have you got. So it's going to be all right. You've said ATP synthase. You've said water is produced. You've said a condensation reaction. You've mentioned the composition of ATP, adenine, pentose sugar, 3 phosphate molecules. So you've really covered everything about what ATP is made of and how it forms. And that's going to get you your 4 marks. All right, let's move on. Please place a tick in the appropriate box next to the equation which represents the reaction catalyzed by ATP synthase. Okay, so ATP synthase. We've got all these different reactions here. We've got ATP going to ADP, PI, and water. We've got ATP plus water goes to ADP plus PI. We've got ADP plus PI goes to ATP and water. ADP, PI, and water go to ATP. So which is the correct one that ATP synthase does? So we've got to look at the word synthase. What does synth mean? Think of synthesize, photosynthesize. What does synthesize mean? Well, synthesizes means making or creating. So ATP synthase synthesizes or plays a role in the synthesis of ATP, in making ATP. So we're really looking for an answer where ATP is on the right hand side because it's being made. So these top 2 are definitely wrong. So it's got to be one of these bottom 2. Okay, because ATP is being made in both of these. However, This bottom one is showing that water is used in the making of ATP. Actually, water is released in the making of ATP, so it's this third answer here is the correct answer. Remember, it's a condensation reaction that forms bonds and will join these together. Biological molecules, whenever we put small molecules together and we join them together, they are condensation reactions and they release water. When we want to break molecules, we need to use water, and those are hydrolysis. So the answer here is the third one down. Awesome. All right, guys, make sure you hit the like. Let's move on. The new antibiotic, so don't know what this question was about, but it's something about an antibiotic, is safe for humans to use because it doesn't inhibit ATP synthase found in human cells, right? It doesn't get in the way of making ATP. Suggests why human ATP synthase is not inhibited and bacterial synthase is inhibited. Okay, so we haven't got all of the question here, but essentially they're saying what could be the difference between human ATP synthase and bacterial synthase that means antibiotics don't disturb humans' production of ATP if you take the antibiotics? So what we have to remember is that bacterial synthase is an enzyme, ATP synthase is an enzyme. So this is an application question. You've got to get your head down and think, okay, they're asking me about enzymes. What's the difference between any two enzymes? So what is the difference between any two enzymes? And the difference is, of course, I know you know it, they're going to have a different tertiary structure. Of course, they'll have a different primary structure, which informs the tertiary structure. So they'll have a different 3D shape. Which bit are we interested in? We're interested in the active site of the synthase molecule. This means that the bacterial synthase is going to be different than human ATP synthase. The active site will be different. And this means that the bacterial synthase can be inhibited, but the inhibitor is not complementary to the human ATP synthase. So what they're really getting you down here is knowing that they're both enzymes, so they're going to have different shaped active sites. The human ATP synthase is not complementary to whatever's inhibiting the bacterial synthase. So it's only 1 mark. So if you just said they have different shaped active sites, they have different 3D structures, hence different shaped active sites, or human ATP synthase, the active site is not complementary to the inhibitor. These will get you the mark. Okay, so that was a bit of a weird question. Let's move on. Okay, moving on. ATP is an energy source used in many cell processes. Think photosynthesis, respiration, those kind of things. Give 2 ways in which ATP is a suitable energy source for cells to use. So why is ATP a good source of energy? Well, it's a small molecule and it can move through the membrane of a mitochondria. So that's important because, of course, we produce lots of ATP inside the mitochondria. It's also, because it's a small molecule and only releases a tiny bit of energy when it breaks down to ADP plus PI, this means it doesn't produce too much heat, so we don't get a lot of wasted energy, okay? Because each ATP molecule just generates a little bit of energy, and so we don't get too much overheating going on. Also, one of the most important things with ATP is it can be used to phosphorylate other molecules. Think of your steps in the Calvin cycle when it's used to phosphorylate GP into TP and things like that. Why is phosphorylation good? Well, phosphorylating molecules, adding phosphate groups to them, makes them more reactive. Okay, so makes them more likely to undergo the reactions we need those molecules to undergo. So that's a good thing about phosphorylation. What else can we say? That's about it, really. We only need 2 marks. So we've said it's a small molecule. It can get through the membrane, releases energy, and it's good for phosphorylating other molecules and making them reactive. Okay, moving on. Describe how ATP is resynthesized in cells. Pause the video, have a go. So, yeah, the enzyme ATP synthase is involved in making ATP. Remember, that's the one that synthesizes ATP. What it does is a condensation reaction where water is released. So these are all facts we can put down. Again, it's a describe question, so just reel off the content. So enzyme ATP synthase carries out a condensation reaction. And what 2 molecules does it join together? Well, it joins together adenosine diphosphate, you can abbreviate to ADP, and inorganic phosphate, you can put capital P, lowercase I. And really, that's going to get you all your marks. So that's how we resynthesize ATP. We join ADP and PI together using ATP synthase. Boom. Forgive me if the ATP questions are sounding awfully familiar, but give two ways in which the hydrolysis of, or hydrolysis, however you want to say it, of ATP is used in cells. So how do cells use the breaking of ATP? We just went over this a couple of questions ago. So what is it that when we split ATP, what is it that we produce that is useful for cells? Make sure you pause the video and have a go. All right, let's talk about those answers. So, of course, when we break ATP down, it turns into ADP and PI. The inorganic phosphate can be added to other molecules, and we call that phosphorylation. And when it's added to other molecules, it makes those molecules more reactive. So make sure you state that phosphorylation of other molecules makes them more reactive. So we don't just use ATP to release energy, we use it for phosphorylation as well. So yeah, and of course, when ATP forms ADP and PI, it releases a small amount of energy that can be used for many different processes. I'll give you an example, chemiosmosis. So when we want to actively transport hydrogen ions across the membrane of the mitochondria and the cristae, we want to transport those hydrogen ions. There we go. We can use ATP. If you think about the chloroplast in the photosynthesis reactions, in the light dependent reaction, we need to transport those hydrogen ions across the thylakoid membrane as well, and that uses active transport, which requires molecules of ATP. So ATP provides energy for lots of different processes in the cell, and you could give an example, for instance, the active transport of hydrogen ions. You could talk about mass loading or active loading in the phloem, where we do the active transport of sucrose molecules in order to create a lower the water potential at the top of the phloem for mass transport, where water molecules move in by osmosis after that. You wouldn't need to go into all the details, but you could name a process which ATP is involved in. And there we go, we get another mark. Wonderful. All right, guys. So if you like the video, it's a small topic today, but if you like the video, share, like, subscribe. Good luck with those exams. Keep working hard. Let's get to the marks.
You're still here? It's over.